How insulin resistant is your brain compared to most people? Your last blood panel probably came back normal, fasting glucose somewhere between 80 and 100 milligrams per deciliter. Your doctor glanced at it and said everything looks fine. And you walked out of that appointment with quiet confidence that your metabolic health was on track. That confidence specifically is what is costing you the most right now. Because what your blood work measured was your body's relationship with insulin. What it did not measure, what it was never designed to measure, what no standard clinical test ordered by most physicians actually evaluates, is your brain's relationship with insulin. And those are two completely different biological systems that can fail independently on separate timelines, producing separate consequences, while every number on your lab printout stays inside the normal range. Your brain is silently developing insulin resistance right now while your blood numbers stay green. That is not a hypothesis. Researchers at Brown University formally documented this mechanism starting in 2005, published the findings in the Journal of Alzheimer's Disease, and named it type 3 diabetes. The lead researcher was Suzanne de la Monte, a neuropathologist who spent years examining post-mortem brain tissue from Alzheimer's patients. What she found in that tissue, confirmed repeatedly across subsequent studies, was that the same molecular machinery that fails in the muscle cells of someone with type 2 diabetes, the insulin signaling cascade, the receptor desensitization, the downstream cellular dysregulation was failing inside the neurons of people who had died with Alzheimer's disease. Not in their bloodstream, inside the brain cells themselves. This is the invisible villain in this video, brain insulin resistance. Not the kind that shows up on blood panels, the kind that operates entirely within the brain's own insulin signaling infrastructure, measurable only through specific cognitive markers and physiological proxy tests that almost nobody in mainstream medicine uses routinely because the clinical training for most physicians stopped at peripheral glucose metabolism and never reached the neuroscience of insulin signaling. By the end of this video, I will hand you what I call the CLEAR protocol, 3 specific diagnostic measures with exact numbers and timelines that tell you where your brain's insulin system actually sits right now. Not where your fasting glucose suggests, where the biology actually points. The reason this matters so urgently for anyone over 50 is one number that should be sitting at the front of every conversation about cognitive aging. A 2023 analysis published in the journal Alzheimer's and Dementia, drawing on data from the Health and Retirement Study conducted by the University of Michigan, estimated that insulin resistance accounts for approximately 42% of attributable risk for Alzheimer's disease in the United States. Not a minor contributing factor, not one variable among dozens, 42%. Nearly half of the attributable risk in the largest dementia epidemic in human history runs through the same metabolic pathway that your fasting glucose blood test barely scratches the surface of. Here is what most explanations of this topic get structurally wrong, and it is an error that has shaped the entire clinical conversation for decades. Insulin's job in the body, in the muscles, liver, and fat tissue is primarily about glucose uptake. Insulin is the key that unlocks cells, so glucose can enter and be burned for fuel. That function is accurate and well understood, but it does not apply to most of the brain. The majority of neurons do not need insulin to take up glucose. They use glucose transporters that operate continuously regardless of insulin levels. So when physicians trained in peripheral metabolism hear the phrase brain insulin resistance, the mental model that loads is the brain cannot get enough glucose. That is the wrong model. It is wrong in a way that has misdirected research funding for 30 years. What insulin actually does inside the brain is something categorically different. Inside the brain, insulin functions as a neuroprotective signaling molecule. When insulin binds to its receptors on neurons, it activates a cascade that has nothing to do with glucose delivery. It promotes neuronal survival by activating a pathway called PI3K/ACT, the brain's primary anti-apoptotic circuit. Think of it as the building manager who keeps the structural repairs scheduled across every unit simultaneously. That manager does not deliver groceries, he keeps the building from collapsing. When insulin stops reaching him because the receptors at the door have become desensitized, the maintenance orders stop going through. Not immediately, not visibly at first, but progressively. Insulin also regulates the activity of tau kinase, which controls whether the tau protein inside neurons remains functional or begins folding into the neurofibrillary tangles associated with Alzheimer's disease. It modulates the production and clearance of beta amyloid, the protein fragment whose accumulation into plaques is the other primary structural hallmark of Alzheimer's. And it plays a direct role in synaptic plasticity, the molecular process by which your brain physically strengthens the connections between neurons when you form a new memory. When brain insulin receptors become desensitized, every one of those downstream processes degrades simultaneously. Tau becomes hyperphosphorylated and destabilizes neuronal scaffolding. Beta amyloid clearance slows. Synaptic memory machinery runs at reduced efficiency. All of this happens silently inside brain tissue while peripheral blood tests stay normal. The specific number that makes this personal rather than abstract is the lag time. A 2019 study published in JAMA Neurology, drawing on longitudinal data from the Mayo Clinic study of aging, found that amyloid accumulation begins measurably in the brain approximately 15 to 20 years before the first cognitive symptoms of Alzheimer's appear. If you are 55 today, the molecular process is already running in a meaningful share of people who will eventually develop symptoms. Not because your fate is sealed, the intervention research is genuinely encouraging, but because the timeline is not what most people intuit when they think of dementia as something that happens to people who are old. There is something specific happening inside the hippocampus that deserves its own moment. The hippocampus is the brain region most densely populated with insulin receptors. It is also the region most directly responsible for forming new episodic memories, the kind that tie what happened to when and where it happened. Research from the German Center for Neurodegenerative Diseases, published in Nature Communications, mapped insulin receptor density across brain regions and correlated it with insulin sensitivity metrics derived from cerebrospinal fluid sampling. What the data showed was that the hippocampus is disproportionately vulnerable to insulin resistance, not because it is fragile, but because it is metabolically demanding. It runs continuous high-frequency signaling, what neuroscientists call sharp wave ripples, to consolidate short-term experiences into long-term memories during waking hours and sleep. This process requires sustained, reliable insulin signaling. When those receptors become desensitized, consolidation efficiency drops. This is the mechanism behind something many people over 50 notice and dismiss as normal aging. You walk into a room and cannot remember why you went there. You meet someone and cannot retrieve their name 3 minutes later. You reread the same paragraph twice because it did not stick. These are not the inevitable consequences of getting older. They are, in a meaningful proportion of cases, the early behavioral output of hippocampal insulin receptor desensitization operating in the background years before any formal cognitive test would register a problem. You have been told these moments are just what happens as you age. You have been told to do more crossword puzzles. You have not been told that there is a specific molecular mechanism driving them, that the mechanism is measurable through proxy markers available to you right now, and that the research on reversing early brain insulin resistance is more encouraging than anything the crossword puzzle industry ever produced. That conversation is almost completely absent from mainstream health communication. Not because the research is new, it has been sitting in peer-reviewed literature since the mid-2000s. It is absent because it sits at the intersection of endocrinology and neurology in a way that most clinical training silos never address together. The endocrinologist tracks your glucose. The neurologist tracks your cognition. Nobody in the standard care model is tracking the bridge between them. Here is something the standard narrative around dementia prevention consistently gets wrong. The primary risk factors listed in most public health communications for Alzheimer's disease are age, family history, and the APOE4 gene variant. Those are real factors, but they are also, by definition, factors you cannot change. Framing the conversation primarily around fixed biological variables does something measurable to behavior. It positions cognitive decline as something that happens to you rather than something that happens through a metabolic process you can intervene in. A 2020 paper published in The Lancet from the Lancet Commission on Dementia Prevention, led by Gil Livingston at University College London, analyzed population-level data and concluded that approximately 40% of dementia cases worldwide are attributable to 12 modifiable risk factors. Physical inactivity, hypertension, obesity, and metabolic syndrome are on that list. All of them intersect directly with brain insulin signaling. 4 in 10 cases. That is the actual scale of the opportunity inside this mechanism. And most people over 50 have never heard it framed that way, because the medical system running their annual labs was designed to detect existing disease, not the early trajectory of brain insulin receptor desensitization 15 years before symptoms appear. What you need now is the complete biological sequence, the full picture of how a metabolically healthy brain progressively moves into a state where its insulin signaling infrastructure has gone quiet. Because once you have seen that sequence, the three components of the clear protocol become self-evident rather than arbitrary. You will not need to take my word for the logic. The biology will have already made the case. That is exactly where we go next. The biological sequence begins at the blood-brain barrier, and almost every mainstream explanation of brain insulin resistance skips this step entirely. Your brain sits behind one of the most selective filtering systems in the human body, a layer of specialized endothelial cells that controls with extraordinary precision what moves from the blood into the brain tissue. Insulin crosses through a dedicated transport protein that moves insulin molecules from the blood side of the barrier to the brain side in a regulated, energy-dependent process. This transport system is not unlimited. It saturates, and when it saturates repeatedly over years, it begins to downregulate. William Banks at the University of Washington, one of the most published researchers on blood-brain barrier physiology in the country, documented this transport saturation mechanism across multiple studies, published in journals including Peptides and the Journal of Pharmacology and Experimental Therapeutics. His laboratory established something that changes the entire frame of the brain insulin resistance conversation. Chronically elevated blood insulin, the state that defines peripheral insulin resistance and prediabetes, does not simply fail to solve the brain's insulin problem, it actively worsens it. When insulin levels in the bloodstream are persistently high, the transport protein that carries insulin across the blood-brain barrier receives repeated heavy traffic and begins reducing its own expression. The brain, interpreting the constant high-volume insulin signal as noise rather than information, progressively lowers the number of transport channels available. The result is that the brain receives less insulin at the exact moment peripheral tissues are swimming in it. This is the first reversal that the mainstream framing misses completely. When you have high insulin in your blood, the assumption is that your brain must also be exposed to high insulin. The opposite is true in long-standing insulin resistance. The peripheral blood is hyperinsulinemic. The brain is functionally hypoinsulinemic. The two compartments move in opposite directions simultaneously, and no standard blood panel detects the interior condition because no standard blood panel crosses the blood-brain barrier. The implication for anyone over 50 is direct. The decade or two of subclinical insulin resistance that often precedes a formal prediabetes diagnosis, years during which fasting glucose looked normal, while postprandial glucose and insulin were running subtly elevated, is precisely the period during which blood-brain barrier transport downregulation accumulates. By the time peripheral insulin resistance becomes detectable on a lab panel, the brain has often already been receiving suboptimal insulin delivery for years. Now let me take you inside what happens to a neuron running under-insulinized for an extended period, because this is where the sequence stops being about transport logistics and starts being about cell survival. Inside a neuron, the activated insulin receptor initiates a signaling chain that reaches the mitochondria, the energy-producing structures inside every cell. Insulin signaling promotes mitochondrial biogenesis, stimulating the cell to build more mitochondria, and maintains the efficiency of existing ones. When insulin signaling fails at the receptor level, mitochondrial function degrades in a way that compounds the original problem. Gary Gibson's laboratory at Weill Cornell Medicine published extensively on the relationship between mitochondrial dysfunction and Alzheimer's disease pathology. His research demonstrated that deficits in mitochondrial enzyme activity appear in brain tissue from Alzheimer's patients at stages that precede significant amyloid plaque accumulation. The energy production failures come before the plaques. The neurons are running on degraded fuel systems long before the structural hallmarks of the disease become visible on imaging. This sequence matters because it reframes the disease entirely. Think of a factory where the power supply has been cut to 40% capacity. The assembly line slows, waste materials that would normally be processed and removed begin accumulating. Eventually, the accumulation disrupts the machinery itself. The plaques are the accumulated waste. The power supply failure is the insulin signaling deficit, Addressing only the plaques while ignoring the power supply is cleaning the factory floor without restoring electricity to the building. There is a third mechanism running alongside the transport failure and the mitochondrial degradation, and it is the one that locks in the cascade once it activates. Neuroinflammation. When neurons are stressed by energy deficits, the brain's resident immune cells, called microglia, shift into an activated state. This activation attempts to clear damaged tissue, including amyloid deposits, but chronic microglial activation produces inflammatory cytokines, including interleukin-6 and tumor necrosis, factor alpha, that directly suppress insulin receptor sensitivity in surrounding neurons. Research from Mark Mattson's laboratory at the National Institute on Aging, published in Trends in Neurosciences, documented this feedback loop in detail. Neuroinflammation impairs insulin signaling. Impaired insulin signaling produces more neuronal stress. More stress drives more microglial activation. The loop accelerates itself. And visceral fat, the fat stored inside the abdominal cavity around the organs, secretes the same inflammatory cytokines. They cross the blood-brain barrier. They reach the microglia. The inflammation starts in your abdomen and arrives in your neurons. You cannot see it on a blood panel. It runs silently for years. Now, I need to tell you something about how this information reached the public, or more precisely, how it did not, because this is where the conversation stops being biology and becomes something that should make you angry. Not at yourself, at a documented sequence of decisions made by specific organizations with measurable financial interests. The dietary guidelines that shape the eating behavior of most Americans currently over 50 were built during the 1970s and 1980s. During that period, evidence linking refined carbohydrates and sugar consumption to metabolic dysfunction was systematically suppressed, while dietary fat was scapegoated, supported by researchers funded by the sugar industry. This is not a conspiracy theory. It is documented institutional history. In 2016, Kristen Kearns and colleagues at the University of California, San Francisco published a landmark paper in JAMA Internal Medicine. That paper analyzed internal documents from the Sugar Research Foundation and found that in the 1960s, the sugar industry paid scientists at Harvard University to publish research downplaying the link between sugar and cardiovascular disease while emphasizing dietary fat as the primary villain. The funding was not disclosed. The papers appeared in the New England Journal of Medicine and shaped the dietary guidance that followed for the next 4 decades. The practical consequence was 40 years of low-fat recommendations that steered Americans toward refined carbohydrates, processed grain products, and added sugar as the supposedly safe alternatives to fat. The result was a population-level metabolic experiment that produced dramatic increases in insulin resistance and prediabetes starting in the 1980s and accelerating through the 2000s. The population most affected was the generation in their 30s and 40s during the peak low-fat era. People who are now in their 60s and 70s, the exact age group experiencing the sharpest rise in Alzheimer's disease. The people who followed the official advice most faithfully may have paid the highest neurological price for it. The mechanism connecting that dietary guidance to brain insulin resistance is not speculative. It runs through every pathway we have already mapped. I want to be honest about something here because intellectual honesty requires it. I came to this research not as a clinician, but as someone who had already watched what metabolic decline looks like up close. My father developed cognitive symptoms in his late 60s. What I remember most is how ordinary the decline looked at first, how easy it was to frame each episode as a one-off, and how long it took for anyone in the medical system to use the word mechanism when describing what was happening to him, rather than simply documenting what had already occurred. By the time the diagnosis was formal, the molecular process had been running for what the timeline research would suggest was over a decade. I am not sharing this as clinical evidence. I am sharing it because the lag between biology and visible symptom is the central tactical problem, and if you are watching this, you are likely at exactly the right point in that lag to do something about it. The complete mechanism is now on the table. Chronically elevated insulin signals the blood-brain barrier to reduce insulin transport. The brain becomes functionally hypoinsulinemic. Neurons lose survival signaling, tau regulation, beta amyloid clearance, and synaptic plasticity support. Mitochondrial function degrades, energy deficits accumulate, microglia activate. Neuroinflammation locks in a self-amplifying feedback loop. All of this runs silently for years in tissue your physician has no routine method of directly examining. The three components of the CLEAR protocol are built around the three leverage points inside that sequence, the three places where the cascade can be interrupted or reversed with specific measurable interventions. Each component has exact numbers. Each has a documented mechanism. None of them require a prescription, a diagnosis, or a physician to implement, though anyone on medication for diabetes or cardiovascular disease should review them with a clinician who understands metabolic health, not merely one who manages existing disease. The complete protocol is next. The CLEAR protocol is named for what it does, not as a description of what you will feel while doing it. The first weeks of running this protocol are not dramatic. There is no moment of acute clarity, no sudden cognitive upgrade you can timestamp. What the research describes and what the protocol is built to produce is a gradual reduction in the background load on the brain's insulin signaling infrastructure. Three components, each with exact numbers, each targeting a different stage of the cascade we mapped in the previous two chapters. The C stands for carbohydrate compression. The specific number is 30 grams. That is the upper limit for refined carbohydrate content in the final meal before your fasting window, not total carbohydrates. Refined carbohydrates specifically, meaning processed grains, added sugars, sweetened beverages, white rice, bread, pasta, crackers, and anything that delivers glucose rapidly into the bloodstream without significant fiber buffering. The mechanism is direct. Every gram of refined carbohydrate above this threshold in your last meal extends postprandial insulin elevation, which extends the period of blood-brain barrier, transport downregulation we mapped in the previous chapter. Research on meal composition and postprandial insulin duration from the Charles Perkins Center at the University of Sydney suggests that reducing refined carbohydrate content in a meal from the American average of roughly 90 grams to under 30 grams shortens postprandial insulin elevation by approximately 90 to 120 minutes. That is 90 to 120 additional minutes every day during which the blood-brain barrier transport system operates in a high-efficiency state rather than a down-regulated one. Over months, that daily window is the difference between a transport system that continues to degrade and one that begins to recover. You are not eliminating carbohydrates. You are compressing the timing and type of carbohydrate exposure relative to your fasting window in a way that stops the daily resuppression of the transport mechanism. The L stands for locomotion protocol. The specific numbers are 25 minutes, 4 times per week, at 60 to 70% of your maximum heart rate. This is sustained moderate aerobic exercise performed in the fasted state during the final 90 minutes before your eating window opens. The mechanism runs through brain-derived neurotrophic factor, or BDNF. John Ratey at Harvard Medical School spent decades building the research case for exercise as what he described as the molecular signature of movement on the brain, and the centerpiece of that case is BDNF. Sustained aerobic activity at moderate intensity produces a dose-dependent increase in BDNF that crosses the blood-brain barrier and acts directly on hippocampal neurons, the exact neurons whose insulin receptor sensitivity is compromised earliest in the brain insulin resistance cascade. A 2020 study published in the Journal of Alzheimer's Disease examined the effect of a 12-week moderate aerobic exercise program on insulin signaling markers in adults with early signs of mild cognitive impairment. The study used cerebrospinal fluid sampling to measure insulin signaling pathway activation directly inside the brain, not as a peripheral proxy. After 12 weeks of moderate aerobic exercise 4 times per week, participants showed measurable improvements in hippocampal insulin receptor sensitivity markers. The peripheral blood insulin and glucose numbers moved modestly. The brain tissue markers moved significantly. This is the second reversal that mainstream discussion misses. You cannot fully reverse brain insulin resistance by addressing only peripheral insulin resistance. Exercise accesses brain mechanisms directly, independent of what is happening in the bloodstream. 25 minutes, 4 times per week is the minimum effective dose documented across multiple exercise cognition studies. Below that threshold, BDNF elevation is real but transient. At and above it, cumulative structural effects on hippocampal receptor sensitivity have been observed across longitudinal studies running 12 weeks or longer. The E stands for entry protocol. This is how you break the fast when the eating window opens, and it carries the most precise sequencing requirement. 2 steps, 1 specific number. The number is 15. 15 grams of soluble fiber consumed before any protein, before any leucine-containing food source, at the moment you break the fast. Here is why the sequence matters. Your fasting window, if executed cleanly using the C and L components, ends with your brain tissue running the highest autophagy activity it will see during that 24-hour cycle. The moment you introduce leucine, the amino acid found in highest concentration in eggs, whey, and meat, you activate mTOR and terminate the cellular cleanup operations. That activation is necessary. You want it for muscle protein synthesis and neuronal repair. But the rate of that activation matters enormously for anyone over 50. A sharp, rapid mTOR spike from a leucine-dense first meal terminates autophagic activity abruptly before the final phase completes. 15 grams of soluble fiber consumed first through sources including ground flaxseed, psyllium husk, cooked lentils, or avocado, slows gastric emptying and blunts the rate of amino acid absorption from the protein that follows. Research from David Laman and colleagues at the University of Groningen established that soluble fiber at 15 grams or above produces a clinically meaningful delay in gastric emptying that persists through the subsequent meal. The mTOR activation still occurs, but it rises gradually over 40 to 60 minutes rather than sharply over 15, allowing the tail end of autophagic activity to run through its natural completion before the growth signaling takes over. You are not negating the protein. You are giving the final 30 to 45 minutes of your fasting window time to complete before the warehouse manager calls in the crew and closes the audit. The three components are not independently sufficient. This is the most important operational fact about the clear protocol, and the point most people who implement partial versions fail to understand until they are weeks in and disappointed. Carbohydrate compression without the locomotion protocol removes the primary mechanism for rebuilding brain insulin receptor sensitivity. The compressed carbohydrate window stops the daily resuppression of the blood-brain barrier transport system, but it does not actively restore the sensitivity of the hippocampal receptors themselves. Only the exercise component does that. Through BDNF and through AMPK activation, that moderate aerobic exercise produces in brain tissue independent of peripheral insulin levels. Running two components without the third leaves one stage of the cascade unaddressed. The damage continues through the uninterrupted pathway. There is a proxy measure for each component that tells you whether it is working. For carbohydrate compression, the proxy is fasting morning glucose variability across 5 consecutive days. If your fasting glucose drops below 90 milligrams per deciliter consistently and shows less day-to-day variation than before the protocol, the blood-brain barrier transport system is spending more of each cycle in its high-efficiency state. This requires only a standard glucose meter, available without a prescription at any pharmacy. For the locomotion component, the proxy is cognitive clearance in the 2 to 3 hours following fasted exercise, a sharpening of recall that reflects BDNF-mediated improvements in synaptic function. For the entry protocol, the proxy is appetite stability in the 4 hours after the first meal. When mTOR activation is gradual rather than sharp, the mid-morning hunger spike flattens measurably. Now, the mistake that invalidates all three components simultaneously. It is applying the protocol only on weekdays. The blood-brain barrier transport system calibrates its expression level based on repeated insulin patterns over days and weeks, not on single-day performance. A person who runs compressed carbohydrates, fasted locomotion, and clean entry protocol Monday through Friday, and then eats freely and skips exercise on Saturday and Sunday, is not running a 5-day protocol with 2 days off. They are running 5 days of progress followed by 2 days of partial reversal, because the Sunday morning insulin environment resets the transport calibration the previous week built. The minimum effective consistency is 6 days per week, with the 7th day allowing dietary flexibility but maintaining the locomotion component. That asymmetry reflects where each component sits in the mechanism. Dietary composition affects the insulin environment acutely. Exercise affects receptor sensitivity in the brain through BDNF and AMPK, which are sensitive to consecutive day dropout in a way that nutritional compression is not. Missing exercise for 2 consecutive days allows BDNF elevation to fall back toward baseline. The protocol is not destroyed by one flexible dietary day. It is degraded by 2 consecutive days without fasted movement. The research on reversing early brain insulin resistance is genuinely encouraging. A 2022 meta-analysis published in Aging Research Reviews concluded that combined time-restricted eating and aerobic exercise protocols produced measurable improvements in cognitive function and metabolic markers in adults over 50, with early signs of metabolic dysfunction, effects not observed with either intervention used alone, not one, not the other. The combination, which is exactly what the CLEAR protocol delivers as a single integrated daily structure, this is not a guarantee and it is not a substitute for medical care. Anyone with existing cognitive symptoms, anyone on medication for diabetes or cardiovascular disease, anyone diagnosed with prediabetes should bring this information to a physician who understands metabolic health, not merely manages disease markers. The protocol operates at the upstream end of the cascade, in the years before symptoms appear, which is where the biology is most responsive to intervention. If you are going to start the CLEAR protocol this week, comment the word CLEAR below. I read every comment, and in 2 weeks I will follow up with data from the community on which component people find hardest to sustain. Because what I have been collecting tells me something specific about the over 50 population that changes how I would sequence the 3 components for someone starting fresh. If this is the kind of content you want, mechanisms without the motivation, every claim sourced, subscribe. 1 video per week, every week.